welcome to my poster, my fellow plant lovers. My name is Mimi. I'm a master's student in Dr. Kevin Simonin's Environmental Plant Physiology Lab at San Francisco State University. This poster is all about my project, tracking leaf trait differentiation of the newly diverging subspecies Chenopodium oahuensis on the Hawaiian Islands. If you look to the left, we have a map of all of my different populations. Um, all of these populations receive direct sunlight exposure. The islands of Oahu, Molokai, and Maui all receive saltwater spray, but the populations that reside on the island of Molokai actually live in sea cliffs and not on the beach. The last site we have represented here is on the Big Island, which lives in a subalpine habit above the cloud line. All 6 different populations, we will be measuring gas exchange using a Liquor 600 leaf perometer. We will be measuring turgor loss point and osmotic potential using pressure volume curves. We will also be tracking leaf morphology by measuring leaf area and leaf thickness. And we'll be counting stomata density and size. Our main objective is to track how these different populations are changing their water use strategy to adapt to these slightly different environments. The bottom half of my poster, I have displayed preliminary data and preliminary results. Please note that data collection and data analysis is not yet complete, so all of this is just a sneak peek of what's going to come. We have 6 colorful boxes in the center here representing our 6 different populations. Please refer to these colors when you're trying to figure out what population that you're looking at on all 4 of these graphs. On the top left-hand corner, we have maximum stomatal conductance plotted on a violin plot. We calculated this using stomatal density and stomatal size. The bottom left-hand corner, we have observed stomatal conductance that we physically measured on the most recently matured leaf using that Liquor 600 perometer that I talked about earlier. Please note that the Elio point population on the island of Molokai has the highest potential for stomatal conductance. On the top right-hand corner, we are comparing turgor loss point between all of the populations. As you can see, our Big Island population is significantly different from all of the different sites. Our Elio point population is trending to be significantly different from the Maui and Oahu populations. On the bottom right-hand corner, we have leaf lamina area and leaf thickness being compared. Leaf lamina area is on the x-axis and leaf thickness is on the y-axis. All of our Molokai populations are trending towards a smaller, thicker leaf, while the Big Island, Oahu, and Maui populations are trending towards larger, thinner leaves. These changes in leaf morphology and plant physiology are super exciting to see even within these first couple weeks of growth, and we're expecting these traits to diverge even more as these plants move towards flowering adulthood. At the end of my project, I will be donating seeds that we collect to restoration efforts on the Hawaiian Islands. As you know, with climate change happening, sea levels are rising, and a lot of these sites are in danger of degradation or disappearing completely. My research will help inform what sites need to be prioritized given our current climate projections. Thank you so much for visiting my poster.